In the previous lesson on DNA, we learned that DNA can be duplicated. And this is important when a cell is going to reproduce. It will duplicate its DNA so that each daughter cell receives a copy of the duplicated information. Now, as it happens, the process of DNA duplication can produce errors. And these errors are called mutations. And this creates variation in DNA sequences in the offspring. And to see how these errors can happen, we'll take a look at our animation again. So here we have one of the strands of DNA, and here we have DNA polymerase, and it's building the missing strand. So let's let it play here. Now again, remember, due to the pairing rules, and certain bases are sticky with others, we're going to have most often then the appropriate base will be paired up with its partner. But sometimes the wrong letter gets inserted into the growing strand. And so DNA polymerase here is not perfect. Sometimes it will stitch together the wrong base. You'll get a mismatch. And that means that we have a change in the DNA sequence at that location. So if there are no mistakes, then the DNA molecule up here at the top will be identical to the one down the bottom here. DNA will be duplicated with no errors. But if there are errors, there will be a mismatch of letters along one or the other strand here, possibly both, in which case then you no longer have identical DNA molecules. So DNA polymerase, as it makes its way along the template strand of DNA, it stitches together the new strands, but as it does so, errors can happen. Now, the DNA polymerase also performs a proofreading function, checking and correcting mismatched letters, but the system is not perfect. The result is an error rate of one mistake every billion letters copied, but there are billions of DNA letters, and so it's likely that every time DNA is duplicated, there will be mistakes. Now, what does a mismatched pairing mean? Well, for example, in our the DNA up top here, it would be the C chemical, the C base, pairing up with an A. Normally the C would pair up with a G, but if it gets inserted here and then stitched into the growing chain, we have now a mismatched pairing, or a T with a G. Now, there is a significance to these mutations. If we recall, our bacteria cell here has thousands of different proteins, and they do all different kinds of jobs. And proteins perform different jobs because they have different shapes. The shape is what allows them to perform some specific chemical reaction. Now, each protein then has its recipe stored in DNA, but mutations are changing that recipe. So in other words, every time there is a change in the DNA, that represents a potential change in the recipe of a protein. So here then we have the process of DNA duplication has a built-in way to create new protein recipes. But this is happening in a random fashion. By random, we mean that the mistake is not purposeful. The change doesn't happen in order to make a better protein recipe. In fact, most often, the change messes up a protein or has no effect. So we need to think of these things as molecules, and they're performing a certain kind of reaction. Now, we do want to say that DNA polymerase has a kind of a purpose or a function or a job. Its job is to duplicate DNA. But by random, what we mean is that the DNA polymerase, when it makes a mistake, the protein itself is not trying to make a certain kind of mistake for the benefit of the cell. That's what we mean by these random errors. So if we think about a protein, of course, it has its recipe stored in DNA, and we'll figure out how the recipe is stored in the next lesson. But if we go changing the recipe, then we're potentially changing the shape of the protein, which means we might be changing its function. And if you change the shape, you can mess up the function. So any change in a protein's recipe is more likely to mess up the protein, because there are many more ways to mess up something than to make it better. Sometimes, though, a change in a protein's recipe can make it do a better job. Maybe it's more efficient at doing this chemical reaction. But most often, changes to the recipe for a protein are harmful in a sense. So the existence of mutations then means that offspring are not identical to their parent or each other. So here we see the parent cell here, and we have offspring with different mutations. So in this case, there would have been an error both in the growing strand up on the top one, and a different error in the bottom strand here. And that would produce then two daughter cells with different mutations. And as a result, they might be making different proteins with different shapes and have different characteristics. Mutations change DNA information, which can change protein shape and function, which can change the bacteria cell as a whole. So because of mutations, bacteria populations can change over time. Such change is called evolution. And we'll study this process of evolution in later lessons, but now we'll just note that evolution refers to changing populations. An individual is not really evolving. This individual inherited DNA and this mutation. This individual inherited the parent's DNA and that mutation, but we don't say that individuals evolve. Populations evolve, and we'll get to the bottom of that in a later lesson. In summary then, DNA mutations are changes in the DNA molecule itself due to the mismatch of pairing of bases with their wrong partners. And this happens every time DNA gets duplicated. There will be some error rate. And these mutations then are changing the information stored in DNA. Those changes most often are harmful or neutral, but sometimes those changes might be beneficial.